Hi everyone, I'm Thomas Keane from the European Bioinformatics Institute, and together with my co-lead Oliver Hoffman, I'm going to provide the update for the large-scale genomics workstream and just give a flavor of what we've been working on the last number of months. Large-scale genomics, so we create standardized methods for accessing large-scale genomic data. We work at a variety of different levels, be it at the reference genome, read data, genetic variation data, expression data, and we work across a variety of different representations. So we work in terms of the files and standards for and formats for storing and genomic data. We create APIs for accessing data over secure connections over the network, and increasingly we're trying to integrate our standards now into cloud platforms, and looking towards sort of more federated analysis models. We're organized into 6 task teams, and we've got a whole network of standards maintainers. So the first group I'm going to provide an update about is our Retrial Format working group. This group is really looking at the ways we store the sort of raw genome sequencing data in terms of the SAM and BAM and CRAM formats. Recent work has included standardizing the representation of base modifications. That's now been included into the SAM and CRAM specifications. We are also providing updates and additions to the compression algorithms used by the CRAM format. These have been incorporated into the CRAM version 3.1 release that happened recently. In terms of genetic variation, the variant call format, the VCF format, this is one of the most widely used formats for storing genetic variation data at scale. We had an incremental release in June, version 4.3. This is really a non-breaking change, improving, clarifying many parts of the spec which were previously ambiguous, and improving the alignment with our SAM and BAM and CRAM formats. There is a lot of ongoing work happening in the VCF standard. There is one particular effort around trying to improve the representation of structural variants. And then there is another group that are trying to look at the scalability of the VCF format, or possibly a successor, in terms of when you start to look at genetic variants from large scale populations into the sort of 50 to 100,000 individuals and beyond, and how the format scales. So there is some evaluations happening in that group in terms of possible alternatives or evolutions of the spec. Another area that we work in is looking at secure streaming by APIs of genomic data, be it genetic variation data or read data. We have the HCS GET API standard, which was adopted by GA4GH a couple of years ago. This year and most recently, this group has been looking at supporting more complex queries. And I think the major focus is really on deployment and implementation of this standard into more of our driver project software stacks, and really trying to get adoption of this standard by the community. Another area that we work in is looking at encryption of genomic data, and in particular, just trying to put an encryption container around our current formats so that we can continue to work in the current file formats that we have, but trying to add some encryption technology around those. So we have developed a standardized encryption container called Crypt4GH. Again, this was a standard that was adopted and approved by GA4GH last year. And again, the focus is really on deployment of the standard and adoption of the standard and implementation. So it now has been implemented in HCSlib, HCSJDK, and a variety of our driver projects have now started to look at adopting and implementing this standard into their platforms. And there will be a breakout session for this in day 2 of the meeting. The next area that we've recently been doing quite a bit of work in is this area of reference sequences, and how do you retrieve reference sequences, and trying to avoid all this confusion we have over human generated labels. And we now have this RefGet standard. Again, this is an adopted standard, I think an improved standard last year from GA4GH, and that was at the individual sequence level. We're now extending this to look at sequence collections, i.e. a genome, and increasingly starting to look at reverse lookup. So how do you go from the human generated labels back to the actual to retrieve the checksum sequence and then to get the actual underlying genomic sequence. And these are really been driven by use cases that are coming from our driver projects as we get sort of engagement with the driver projects for this standard. And then finally, this is a much more recent effort that we've had coming from the RNA task team. They're increasingly starting to look at sequence annotation, thinking about models for sequence annotation, modeling the different types of features, genes, transcripts, and there's been probably decades of work in this area has been carried out by various people in the community. And I think what they're really starting to think about use cases, in particular clinical user stories, trying to gather those, trying to get interest from driver projects, and then trying to build interest in potentially creating a common sort of gene transcript sequence feature annotation model from GA4GH. So this is a very early effort. It's a joint effort with the Genomics Knowledge Standards group, and I would encourage you to engage in this group if this is of interest. So that's the updates for the large-scale genomics workstream, and I'm sure we'll be happy to take any questions at the appropriate point in the meeting.